Concept 13.3, Meiosis. In this concept, we're going to be going over the stages of meiosis and then compare meiosis with mitosis. So, the stages of meiosis. It all begins with prophase, like in mitosis. But meiosis will have two nuclear and cellular divisions. So we call the first prophase, prophase 1. It's nice when the terms make sense. Like in mitosis, the chromosomes begin to condense during prophase 1. Unlike in mitosis, the homologous chromosomes loosely pair along their lengths, aligning gene for gene. Crossing over, or the exchange of corresponding segments of DNA molecules by non-sister chromatids, is completed while homologs are in synapses, held tightly along their lengths. This allows the homologous chromosomes to switch genes. Meanwhile, the nuclear membrane breaks down and the spindle forms. The spindle is attached to the kinetochores. Meiosis skips pro-metaphase and moves directly into metaphase 1, where the chromosomes line up at the equator. Notice that the sister chromatids are still attached. During anaphase 1, the homologous chromosomes are separated. Remember in mitosis that the sister chromatids are separated at this point, but in anaphase 1 of meiosis, the sister chromatids remain attached. The cell divides during telophase 1 and cytokinesis. The resulting cells move immediately into prophase 2. Since the homologous chromosomes are now in separate cells, there can be no crossing over. A new spindle forms in each cell. The spindle is attached to kinetochores, and the chromosomes line up in metaphase 2. In anaphase 2, the sister chromatids separate. Telophase 2 and cytokinesis occurs, and you have 4 total cells. They're different from each other and from the parent cell. They are haploid with 1 copy of each chromosome. Let's compare meiosis to mitosis. In meiosis, you have synapsis and crossing over during prophase 1. In mitosis, there is no synapsis and crossing over during prophase. In meiosis, during metaphase 1, pairs of homologs line up on the metaphase plate. In mitosis, individual chromosomes line up on the metaphase plate during metaphase. During anaphase 1 of meiosis, the homologous chromosomes separate. During anaphase 2, the sister chromatids separate. During anaphase of mitosis, there's separation of the sister chromatids. Well, that should be enough for this concept. And yes, you will need to know all the phases of meiosis and how they compare to the phases of mitosis. So go back and review the recorded lectures from Chapter 12. That should help.